And first of all, I would like to thank the organizer for having me here. It's been a very interesting conference up to now. And more precisely, actually, I'm delighted that Stefan gave his talk before me because he was able to introduce in great details and much better than I would have done, actually, what range expansion are and why they are important. And so what I'm going to do now is to present you a case study of a range expansion where we see a signal of selection, or at least a change in a color polymorphism. And the question we're trying to tackle is whether this change in color is due to selection or whether it could be due to allele surfing. And so this is a work done in collaboration with several students, Sylvain Antoneza, Ricardo Canitz. Samuel is a postdoc in my group and developed the software that we use for doing the simulation work. Arno is also a new PhD student. Reto is a postdoc in Uppsala now. And Alexandre Roulin, we've heard his talk 2 days ago. So range expansion, as we heard, they are extremely common. And their genetic consequences are largely underestimated. I mean, we've been bathing in a world of island models or stepping stone model where everything was at equilibrium between migration and drift, and we just know that for most species inhabited at least the temperature area, but also in the tropics, demographic changes have been occurring. And not only simply population growth, sudden bottleneck, but it's been going along latitudinal gradients. The classical example is the recession of the last, after the last glaciation, just open space for species to invade, and therefore most species inhabiting Europe nowadays were in refugia 20,000 years ago and had to colonize this world. And we can still see in many of the species the signature of this expansion. And one of the things that Laurent Escoffier has been showing, and others, is that this range expansion creates a genetic signature very, very similar to selective signature. And so one of the very difficult issues when we see something that looks like selection is to be able to say that this is really selection and that is not a random neutral drift process going on, or even deleterious alleles being carried out, as we've heard earlier. And so what I'm going to show you is one way to try to disentangle these two processes, selection or drift whilst surfing a range expansion. And so the animal I'm going to use is the barn owl. And well, you see this very conspicuous polymorphism. Some animals are white, some are rufous brown. So this picture is from Switzerland. The two animals live in the same place, and you can find actually sometimes even in the same brood the two morphs. But, and you see that this range of color goes from completely white animals to completely brown animals, okay? And if you look across Europe, you see that there is a clear pattern. It's not, the animals are not randomly located in Europe, but you see that the white animals tend to be located in the Iberian Peninsula, while the rufous brown animals tend to be located in northeastern Europe. Okay, so there is a clear climb there. And this is a case for color, like the white and rufous brown, but this is also a case for spottiness that Alexandre mentioned 2 days ago. So I'm going to focus on only the color, the white to brown color climb. And so to try to tackle these questions, what we did is we sampled several populations across Europe. So we have 20 populations, and you see that the samples are spread out from the Iberian Peninsula all the way to northern Germany and Czechia and these places. And the data we've at hand are not very modern next-generation sequencings. They're old microsatellite, old-fashioned microsatellites. But we have 20 populations and 400 individuals overall, with at least 10 individuals per population. And for each of these individuals, we have characterized the color, and we have the genotype at 20 microsatellite markers. And with this data, what we can do is look at differentiation across populations. And overall, we find a very low neutral differentiation, microsatellite differentiation, 0.01, which is kind of expected for bird. On the other hand, we find quite a large differentiation for color, 35%. More to the point, so this is not randomly placed, it's not some populations having high color and others, but we see a clear climb in color, and you see that the slope here, this is, okay, the slope here for the color climb is much, much stronger than the slope for the neutral markers, okay? And so, at first sight, this looks like selection, and this looks like an ANCOVA type analysis, and the interaction is very significant, okay? And we can do that with other measures of differentiation, like YOSD and these sort of things, and we can also play with the irritability level and the irritability among population, and this is very, very robust, and we still see this differentiation for all kinds of assumptions. Right, so this looks like a selection, but again, we're in this range expansion scenario, and therefore in this scenario, we have to ask the question, can this be due to surfing? Could drift explain this pattern? So we took a two-step approach to answer this question, and the first one is to use approximate Bayesian computation, and I will come back to that in a second, to estimate the demographic parameters of the species. Basically, what we have to do first is to understand how did the species colonize Europe? Did it migrate a lot? Did it have a fast growth rate? When did it start expanding? All these kind of questions. And why do we need that? Is that we then use this data and these inferred demographic parameters in a new range of simulations where we're going to model the color trait and see how likely it is that the neutral color trait with different genetic architecture could arise by chance by these surfing processes. So approximate Bayesian computation in a nutshell. We have observations here, and from it, so these observations are allele frequency, and it's very difficult to compare allele frequency from simulation with allele frequency from observed data. So we have to resort to use summary statistics. So summary statistics would be, for instance, the number of alleles, the heterozygosity, the FST between population, and these sort of things. And at the same time, we run simulations kind of realistic, where we try to mimic the shape of the continent, and we assume that we have stepwise migration and these sort of things, and I will detail this in a second. From these individual-based simulations, we obtain also summary statistics, and we compare the two sets of data, and we find out the sets of demographic parameters that allow to best match the observed statistics. And so for that, we use a program that we developed in the group called Quantinemo, and this is an individual-based simulation program that allows to investigate the effect of selection, mutation, migration, and drift on quantitative traits, but also on neutral markers in a meta-population setting where we have different environment and we can put selection, we can put all these sort of things. It's available on the web. You just type Quantinemo and you will find the web page, and there is a large documentation on how to use this. So, how did we proceed to simulate the Barnold populations? So we have a pixelized map of Europe. The red dots correspond to different samples here. We use the same sampling strategy. We use coalescence simulation because they're very fast, and we're going to run very many simulations to infer these parameters. And we use the same statistics for the observed data and for the simulations. So basically what this coalescence is doing is generating this kind of trees, and the red dots at the bottom correspond to samples. And so we start with a refugium. In this example, it's going to be a refugium located in the southern part of the Iberian Peninsula. And we're going to let the population grow. So this is a demographic simulation first. And depending on the speed of the growth rate, on the migration rate, etc., we will colonize more or less fast the whole of Europe. And once it is done, we take samples corresponding to the actual sample that we took on the field, and we run backward simulations, coalescence simulations. Okay? And you see that the genes scatter all around the space, and then gather back into their original refugium. Okay. So with that, we have very many genes for all individuals, and what we're going to do is we're going to compare that to, we're going to extract from this data summary statistics that we're going to compare to observed data. So our observed data are, as we've seen before, and I'm focusing at the moment only on the microset, trying to reproduce the neutral pattern observed at the microset. So we could use all of the pairwise FSTs and heterozygosity and number of alleles per population, but this would make a huge number of statistics that we could summarize using different techniques. Another solution is to look at the data and to find patterns that are very strong. And the first strong pattern is this isolation by distance. We do find isolation by distance. And so we could use simply the slope of the isolation by distance and the intercept of the isolation by distance slope as two statistics. Another striking pattern is the drop in allelic richness across Europe. So here you have Evora, the sample in Portugal. And as we move away from this sample, we see a very strong decrease in the number of alleles as we move away. Okay? So again, we could use all the points or simply take the slope and the intercept of this relation. So if we take the slope and the intercept, that makes 4 statistics. We run ABC using the whole set of several thousand statistics reduced by using partial least square, or we use this method based on these 4 statistics, and they give roughly the same results. So we had this first idea that there was one refugium in the Iberic Peninsula, but there could be 2 refugia. There could be one in the Iberic Peninsula and one also in the Balkans. Okay? So we ran further simulations with these 2 refugia. We assumed in the first set of simulations that carrying capacity was constant across Europe, but we know that the number of birds decreased as we move northward. So we also ran simulations where we had a decline in carrying capacity. So we had high density in Spain and Portugal, and as we move away northeastward, population size decreases. We had another observation is that the risk of extinction of species of population is higher as we move northeast to the northeast. And therefore, we also had a decline in extinction rate as we move away from Spain. So these were different models that we tried and that we compared and to try to see what was realistic. And the parameter that we are using and that we are the age at the onset of the colonization. So when did the expansion start? The migration rate between neighboring populations, and we have a stepping stone dispersal to the neighboring population. The growth rate of this population, the carrying capacity of its patch, the initial refugium size, and the mutation rate of all microsatellite markers. And so first thing is we run 100,000 simulations for each of these scenarios, got the best results out of each of these scenarios, and then what you can do is compare these different scenarios and see which one is the most likely. And you see here that The most likely scenario is a simple one with one refugium, one carrying capacity, and no plan of extinction. It is closely followed by the one with a declining carrying capacity and one with a declining extinction rate. But the simplest one is the one with one carrying capacity, and so we're going to focus on this one for the rest. And you see really that all of the model with two refugia or with constant population size are rejected completely. So our first impression that we had refugia in Spain and colonization from this refugia is what seemed to be happening. And so once we have done that, we can run a further 900,000 simulation to estimate the parameters. And so what you see here on the top is the graph. The gray line is the prior probability, so basically the space in which we're going to draw the different value of the parameters. And the black line corresponds to the posterior probability, what sets of observation of parameters best fitted the observed data. And on the bottom, you have the table with the value corresponding to the mode of this distribution and the 95% high probability posterior confidence interval. And so all of these parameters, so the start of the colonization occurred 7,350 generations ago, which correspond to, so the generation time has been estimated at 3.6 years. So this sets this at 26,000 years, so just before the last glacial maximum. But, and you see that the estimation of the start of the colonization is not very peaky. The carrying capacity, so the size of each patch is pretty well estimated here, around 200 individuals. We have a very poor estimate of the population growth rate. And this is kind of expected because the colonization occurred in the past. And so the signature of this growth rate is not left very strongly in the genetic data. We have pretty good estimation of the dispersal rate and very good estimation of the mutation rate. And all of these estimates are very coherent with what we know about the biology of the species. I mean, they're very consistent with the population size that we see today, with the mutation rate that has been estimated, with current population growth rate and these sort of things. Okay. So now we have that. And what we're going to do is we're going to use this estimate into a new set of simulations. And again, we're going to use Quantinemo this time, but we're going to use forward-in-time simulations, because now what we want to do is to generate the genetic data and generate the phenotype. Okay? So we're going now to simulate a color trait, and we know very little about the architecture of this trait. So we're going to try several different architectures. And to test whether we have selection going on or not, what we're going to use as a statistic is the difference in slope between QST on the simulation and FST on the simulation, and compare this to what we observed in the real data set. Okay? So next slide is the results, and I'm going to walk you through the steps. So the first architecture that we modeled was a simple one locus two allele system, whereby one allele give you the white color and one give you the red color. And what you see here is the difference in slope between QST and FST observed from the simulation. And what you see at the bottom, the black line here, is the observed difference in slope. Okay? And so you see that this observed difference in slope is centered on the value of 0, and that none of the simulation gave us a value as large as the observed. Okay? So out of the 1,000 simulation that we made, we observed that. We never observed that. A second architecture would be still one locus, but this time with 25 alleles in a continuum. So we have a series of alleles moving from white to redder and redder and redder. And again, you see from these simulations that there is no simulation that gave a difference in slope as large or larger than the observed. Next step is to consider that we have a polygenic trait. And so we went for 25 loci, which is a very large number of loci involved in the expression of the trait, with 2 alleles each. Okay? And so we start the simulation, and we have this trait, and we just then pull together the different alleles to obtain the value for the trait in the individuals. And again, we see in these simulations, assuming complete codominance, no, complete additivity of the alleles, that there is no simulation giving us a difference as large or larger. Okay, so for all these simulations, we use a standing variation in the Iberian Peninsula. So we start the simulation with the Iberian Peninsula field and the current frequency of the phenotype in the Iberian Peninsula. So so this means that the frequency is 10% of the red allele in the peninsula, which is quite low. So one way to favor the surfing hypothesis would be to consider that the red color is recessive. In this case, if the red color is recessive, the phenotype is less frequent, but the frequency of the allele is higher. Okay? And so the next set of simulations are with a recessive allele, either at one locus, and again, we see that we have no simulations giving us a difference in slope as large or larger than the observed, or on the side, it's 25 alleles, 25 loci and 2 alleles, and we see the same. Okay, so monogenic or polygenic, we observe exactly the same. Okay, so next step, to force surfing, what we did was to consider that there was a mutation appearing on the population which is at the edge of the expansion front. Okay, so in the Pyrenees, we just put a mutation and see whether this is caught up by the surfing whilst the population expands. And this is what we see next. And so in this case, we have one simulation that gave us a result larger than the observed data. So 1,000 out of 1,000 gave us a result consistent with what we observed in the data. Oops. We did this for one locus and 50 allele, and you see that, again, we have one simulation out of the lot, and with 25 loci and 2 alleles. So in this case, we have all loci each have one mutation occurring, and so yeah, one mutation allowing for this being caught up by the wave of expansion. And we see that in this case, we have 3 simulations giving us a result larger than the observed. Okay? So even in these circumstances where we have really the most favorable condition for population expansion and for surfing during this population expansion, we see that in very, very few cases we observe this expansion. So from this, we conclude that using this approach, it's possible to disentangle the effect of selection and the effect of surfing. We showed that surfing is very, very unlikely. However, this approach is very time-consuming. We've been running millions of simulations. And as you've seen, it's not a one-size-fits-all system because you have to know quite a bit about the biology of your species. You have to estimate the parameters, the demographic parameters via ABC, and then you have to run the forward simulation. And we have yet to estimate whether the selection occurring is strong or not. Okay? And so in the perspectives, this is what we're doing. And at the moment, we're just using a toy world, whereby, so this resembles very much what Stephen presented before. We have population started, so it's a 2-dimensional population with 5 patches across and 51 patches along. Carrying capacity can go from 0 to 100 individuals. So when it's red, it means it's 100 individuals. And we have selection going on along there. So at the moment, we're just looking at one locus, 2 alleles. And the little a allele is favorable at this extreme of the range expansion, and the big A allele is favorable here. And so what we do is we simulate both the traits and neutral markers. We measure QST and FST, and we obtain the difference in slope. And we're going to use that to see whether we're able from that to back estimate the frequency, the intensity of selection. So here on the y-axis, on the x-axis, you have geographic distance, but then after what we only present on this panel, the middle and the other panel, is QS is the slope of IBD for QST and FST, so for the trait and the markers, against the selection intensity. And here is the difference in slope. And we took a subset of these simulations to see whether we're able to estimate correctly the selection coefficient. And you see here that this is the pseudo-observed value that we try to estimate using an ABC approach. And we see that the estimation of this selection coefficient is pretty good in this case, whereas if we have several alleles, the estimation is not as good. And so to sum this up, at the moment, if we have in the true model very strong selection, we can estimate that the selection is very strong. If we have strong selection, we can estimate most of the time, but sometimes we find out, we estimate that the selection is either weak or very strong. And as we go down, we see that the worst situation is when selection is weak, because very often we find out, we estimate that this is a neutral surfing, or sometimes that this is very strong. And when it's neutral, we sometimes do find out that there is selection going on. Okay, so thanks to Vitality, the High Performance Computing Center in the University of Lausanne, and I'm also a member of the Swiss Institute of Bioinformatics and NSF for support, and you for your attention.